Okay, Assalamualaikum. All right, so we're going to do our lab number 3, so test for proteins. So please be ready for your manual. Okay. All right, so today's experiment is laboratory 3, test for proteins. Okay, before we go further, there what we call introduction in your manuals. Okay, the treatments of protein with a copper sulfate in alkali solution, what we call is a Biuret test. Okay, what happened in this test? It will produce the purple to pink or violet color. Right? For any substance that contain two parts inside of the proteins, which is amino groups and carboxyl groups. So this one kita akan belajar dalam chapter protein nanti, alright. But you all dah learn tentang functional group So one of the example of functional group yang ada dekat proteins adalah kumpulan amino dan juga kumpulan carboxyls. So directly joined together. So free amino acid does not give the positive test. Okay. So kalau hanya amino acid yang ada bond sahaja akan ada results. Which mean that kalau letak hanya monomer amino acid saja dalam tu, awak takkan dapat result. The exact color depends on the proteins involved, but it will be in the blue or red range. Okay. So maknanya test for protein ni dia untuk protein sahaja, dia bukan untuk amino acids. So kalau pakai amino acid, awak tak dapat result dia. Okay. Okay, mostly introduction is more on the paragraph 1 refer to the test 1 ataupun experiment 1. Heavy metals ions such as plumbum and mercury ions denature protein probably by reacting with the thiols groups. So one example of another functional groups is the thiol groups, okay, and carboxylic groups. Okay, ini adalah group yang sama, COH ni, which contribute to the toxicity of the heavy metals ions. Okay. So we use what we call certain acids, such as trichloroacetic acid, to produce the complete denaturation and precipitate. So what is denaturation? Denaturation is a kerosakan pada structure protein disebabkan kerana pemutusan ikatan. Right? So from that we can get the precipitate. So I will explain more on the summarize of the experiment nanti dalam PowerPoint. Okay. Treatment with the trichloroacetic acid is often used to stop enzyme catalase reactions. So we use what we call tannic acid and picric acids. So tannic acid and picric acid is from what we call organic enzymes. Organic acids. Will also denature the protein, but less effective compared with the trichloroacetic acids. Okay. The amino acid molecule contain of an amino groups which act as a base, okay, and the carboxylic function, which behave as an acids, a protein donor, in the presence of acids. The amino acid is converted to NH3 or ammonium ions. Base will convert the carboxylic acid to the carboxylated ion, which is COO negative, okay, charges. The isoelectric point for amino acid is the pH at which both functional groups are in their ionized state. Under this condition, the negative carboxylated anion of the one molecule exert an attractive force on the positive ammonium group of another molecules, and the precipitate will be observed. So what is precipitate? Precipitate is a particle yang dapat daripada eksperimen tersebut. Okay. So proses eksperimen ni lebih kepada kalau contohnya awak ada protein ataupun awak ada susu, for example paling mudah susu susu awak letakkan sedikit jus oren. So bila apa yang berlaku dekat situ, cair susu tersebut akan menjadi partikel-partikel yang halus. That's what we call denaturation. Okay, itu adalah pandangan dia, alright. So objective of the experiment is to observe denaturations of protein by heavy metals, cation and acids. Okay, heavy metal cation kita gunakan plumbum dan sebagainya. Okay, mercury dan sebagainya. Okay, determine the isoelectric point of the cations. So kena kita tengok macam mana keadaan berlakunya proses isoelectric pada proteins. So dalam kes ni kita gunakan casein. Casein is one example of proteins. So number one, how to do the Biuret test? The first, you have to pipette 2 ml of stock solution of albumin. So albumin is one example of proteins. Okay, into the test tube and add 2 ml of 0.1 sodium hydroxide and then 5 drop of the 1% copper 2 sulfate solutions. Okay, so kita ada sodium hydroxide. Sodium hydroxide adalah untuk mendapatkan keadaan beralkali dan lain sebagainya. Okay, we have to record the observation and then repeat with others types of solution such as peptone and caseins. Okay, make sure you get the purple to violet color. Then it will be a positive. Number two, test 2 is a precipitate of protein by heavy metals. Okay, so you have to prepare 2 ml of protein solutions. Okay, according with the the picture below. Okay, so you have to do 3 sets of 3 test tube. Okay, albumin. So test label with 1, 2, 3. Okay, but why 1, 2, 3? Because 1, you put the same solutions. Okay, number 1, only you put copper 2 sulfate. Same goes to the casein. Same goes to the peptone. Okay, so 3 sets mean albumin, casein, and peptone. Okay, each of the test tube is going to be added 4 drops of heavy metal solution, which is copper sulfate, mercury nitrates, and lead acetate. Okay, swill the test tube and record the observations. Okay, so this one you have to see if you get the precipitate or not. Right. Add another 5 drop of 0.1 molar copper sulfate, and you have to record the observations. Okay. So quickly go for the test 3. For this one, we're using acid to make sure they are precipitate on a protein. Okay, pipette 2 ml of protein solution, which is albumin, casein, and peptone. So kalau ada 3 sampel, maknanya 3 sets. Okay, so 1 albumin, according with the picture below, okay, we have a 4 test tube in each of solution ataupun sample. You have to label 1, 2, 3, 4. Why? Because we have to put the same acid for 3 sets of proteins. So number 1, you have just put sulfosalicylic acids. Or number 3, only you put tannic acids in each of albumin. Another set will be casein, another set will be peptone. Okay, label number 3 for tannic acid saja. How many? You have to put each of it 5 drop of acidic solution, which means 5 sulfosalicylic acids, 5 drop of picric acid, 5 drop of tannic acid, and 5 drop of trichloroacetic acids. Okay, and then you have to swill your test tube and record the observations. Another one, after that, you have to put 2 ml of 0.1 molar sodium hydroxide in each of test tube slowly and then record observation. So again, this one, test 3 is to see whether present of precipitate or not. Okay, last one is test 4, is isoelectric point of caseins. Okay, first you have to prepare the casein solutions. All right, dissolve 0.1 gram of 1 pellet of sodium hydroxide in 250 ml of distilled water or deionized water. Add 1 gram of pure casein powder. And then you have to stir well until solution is clear. But when you're doing this one, you can get a slightly haziness. Okay. And then quickly do the 2 molar of hydrochloric acids, which is added 4 ml of concentrated acid. Slowly stir with the 21 milliliter of deionized water. Then you will get 2 molar hydrochloric acids. Okay, so this one is prepared by the lab assistant. Okay, because using the concentrated acid. It's very dangerous. And then you have to set up the series of 10 test tube and label them with a 1 to 10. Okay, so apa yang awak kena labelkan, apa yang awak kena letak. Each test tube, you have to label 1 to 10, have what we call pH yang berbeza. Okay, so you just put acetic acid and sodium acetate. Okay, the two things will be prepared by our assistant. So macam mana kita nak dapatkan pH 3.8? We just put according with the protocol. If you put 4.4 ml of acetic acid, 0.1 normality, and then 0.6 ml of sodium acetate, 0.1 normality juga, so you will get around 3.8 pH. To reconfirm, you have to check with the pH meter. Tapi dalam manual ni memang dapat Okay, kalau awak campurkan semua bahan-bahan tersebut dan pH awak akan dapat sama. Right? And then add 1 ml of casein in each of test tube. So which means you must have about 10 ml of caseins. Okay. So setiap satu tube ni 1 sampai 10 ni yang ada kita punya dua bahan tersebut iaitu acetic acid dan sodium acetate, you have to put your caseins 1 ml each, right? And then swill the test tube. Okay, and then record the observation after 30 minutes with the several point. Okay, sama ada dia dalam bentuk colorless ataupun cloudy ataupun precipitate. So setiap test tube tu awak kena recordkan sama ada dia colorless, cloudy atau precipitate. Right? Okay, that's what we call hint in this manual. Okay, so cloudiness will appear but will clear up again as the acid is distributed throughout the system. Eventually, it will reach a maximum as the isoelectric point is achieved. Okay, the system will then begin to clear up as addition of the acid continue, and the casein structure is converted to its acid, what we call protonated form. Okay, once the isoelectric point is reached, as indicated by the failure of the cloudiness to fade. So sekarang ni bila awak buat 10 set of test tube tadi, dia akan jadi daripada failure kepada cloudiness. Okay. So continuity additional of the HCl ataupun acid hydrochlorics will cause the system to clear up again as the pH drop below the isoelectric point. The change can be a reverse back. So kita nak change balik benda tu, kita letak 2 molar of sodium hydroxide. Okay, before the cloudy appeared with persistent at the isoelectric point, then clear as the pH rise by the further additional of sodium hydroxide. Okay, so dekat sini tips dia, awak akan dapat isoelectric point keadaan dia pada pH 4.6. Okay, kalau kita buat eksperimen tersebut so pH 4.6 adalah the best pH for isoelectric point. So keadaan dia adalah seperti dia bukannya clear dan dia bukannya terlampau precipitate, dia lebih kepada cloudy sikit. Right? Itu adalah info daripada kita punya itu itu manual alright. Okay, is there any question before we go for the principle of this experiment? Okay, ada soalan sebelum kita sambung ke principle of this experiment? Okay, please be ready for your notes alright.
Saya nak tanya.
Right, okay. Ya, ada apa ni?
Bila dah sampai dekat isoelectric point tu, dia tambah lagi 2 ml NaOH.
So itu untuk kita kekalkan balik. Pasal asalnya kalau awak dapat casein tu dalam bentuk clear tau. Tapi bila awak letakkan casein tu setiap pada test tube tersebut, dia akan berubah menjadi daripada cloudiness ataupun daripada clear kepada ada precipitate. So kita nak clearkan balik daripada semua tu kepada clear semua, so kita kena letak 2 solution tadi. Faham? So result awak akan dapat clear balik. Okay. So maknanya awak telah berjaya membuat eksperimen tu. Maknanya kita boleh kurangkan denaturation dia. Alright. So further awak boleh tengok dalam video Okay.
Jadi kena kena rekod dua kali benda tu jadilah. Kena lepas dah sampai dekat isoelectric point, rekodkan kata dia jadi coordinates. Lepas tu, lepas tambah NaOH tu, rekodkan lagi lah lepas tu jadi apa?
Okay, yang itu adalah untuk kita apa tu, kita tengok kita recheck balik sama ada dia boleh berubah ke arah clear atau tak. Tapi the first result yang awak dapat tu, yang awak tengok daripada sini, yang ni, okay, selepas awak letak masa eksperimen sebenarnya sampai ke record observation after 30 minutes. Itu saja result awak dapat. Tapi untuk kita clearkan balik pada keadaan asal question tu pada colorless balik, baru kita letak kita punya bahan lain selain daripada kita letak balik ni, bahan ni. 2 molar NaOH dropwise. Tu akan kita clearkan balik semua. Maknanya Isoelectric Point tak akan jumpa lah dekat page tu. Faham?
Faham.
Okay.
Thank you, sir.
Itu cuma kita nak set balik. Tapi kalau the first result tu daripada sampai 30 minit ni kita boleh masuk ke dalam result dah. Sebab kita nak tengok dekat page mana isoelectric berlaku. Okay. Right. Okay, ada soalan lain lagi? Okay, kalau tak ada kita sambung. Okay, kita sambung lagi. Kita sambung pada principle of this experiment. Right. Alright, this one note saya dah bagi dalam future, so boleh jawab dah boleh tengok, boleh download dan juga boleh tengoklah apa dia punya principle okay. So jangan lupa awak jawab awak punya discussion juga untuk attendance today, alright. Okay, before that, what makes proteins? Okay, there are 3 macronutrients, proteins, macronutrients ataupun macromolecule. There will be proteins, fat and carbohydrates. Okay, this one is lebih besar, paling besar sekali. So micronutrients or macromolecules provide calories or energy. Okay. So protein essential in building muscle mass. Okay. So commonly found in animal products. They are also present in other sources such as nut and legumes. Okay, kacang dan juga legumes. Alright. Protein is a basic component of living cells, being made of carbon, hydrogen, oxygen, and nitrogen. So this is the structure inside the proteins, okay, atoms. In the series of amino acids, we call it monomer, and linked by peptide bonds to form polypeptide or polymers. Okay, a protein molecule can have 1 or more polypeptide chains. The 3 types of protein, they are fibrous, globular, and membranes. Okay, awak tengok gambar ni. This is example of the 1 amino acids and 1 amino acids. So bila 1 amino acid tu bergabung, dia akan melepaskan grup mana yang boleh gabung functional group dia. Functional group dia adalah kumpulan karboksil daripada sini dan juga kumpulan amonia daripada sini ataupun kumpulan amin. So amin akan bergabung dengan kumpulan karboksil, melepaskan satu air dan awak akan dapat satu ikatan peptide. So dekat mana ikatan peptide tu? Ni dia, COO, CN and H ni. This is example of one of peptide bond. Okay. So Biuret test, we testing protein sample with the copper sulfate in alkaline solutions. Okay. So positive result with sample contain peptide bond. Okay. So let's say kita ada example of one polymer. Okay. So apa yang berlaku dekat sini? Kita ada copper sulfate. So copper sulfate akan bergabung dengan ikatan yang ada pada 2 protein tersebut. Dua polypeptide tersebut. So apa yang berlaku? Dia akan pergi react kepada peptide bond. Okay, apa yang berlaku dekat situ? Copper 2 blue present in the Biuret reagent, bind to the nitrogen atom at the peptide bonds and undergoes reduction to copper 1 violets. Okay. Since this is the test is not greatly disturbed by the presence of amino acid in the samples, it can be used to gauge the concentration protein in the whole tissue samples. So tujuan utama kita gunakan Biuret test, make sure dia boleh detect ikatan peptide dalam tu. So kalau case amino acid tak boleh sebab dalam amino acid tidak ada ikatan peptide. So kita boleh pakai Biuret untuk amino acid, tapi untuk protein boleh. Sebab copper sulfate akan bergabung dengan kita punya ikatan peptide bond tu. Kat mana? Dekat sumber N tadi. Okay. So what about heavy metals? So heavy metals such as mercury and acids. Okay, for example, trichloroacetic acids or TCA, denatured proteins by reacting the important functional groups that are maintaining the structure integrity of proteins. So what is meaning of denatured? Denatured is affected on the structure is no longer stable. Okay, sebab apa tak stable? Sebab ikatan telah pun terputus. And then you get the precipitate in the solutions. So macam apa masuk solution, apa masuk precipitate is a solution turns colloidal or cloudy. Itu maksudnya berlaku proses denatured tadi. Okay, let's say kita ada folded protein. So kenapa folded protein? Sebab dia ada banyak ikatan dalam ni. Okay, awak akan belajar nanti dalam nota untuk amino acids dan proteins. So setiap amino acid tersebut ada dia punya reaction. So reaction itu akan menyebabkan terbentuknya ikatan. So bila kita putuskan ikatan, berlaku proses denaturation. So denaturation itu adalah berlaku disebabkan kerana unfolding of protein. And then you will get the precipitate. The precipitate dikenali sebagai colloidal ataupun cloudy. Right? Okay, isoelectric point of the proteins or PI. Okay. So isoelectric point is the pH at which both amine and carboxyl groups protein are in their ionized state. Macam mana bentuk dia? Ni kumpulan amin akan mendapat satu charge, kumpulan karboksil akan kehilangan satu H. Okey, itu adalah konsep di sebalik isoelectric. Tapi kalau nak eksperimen awak buat, awak akan nampak cloudy tu. So cloudy tu pada pH 4.6. So pH 4.6 itulah menunjukkan pada isoelectric. So macam mana keadaan dia? So kenapa keadaan tu berlaku? Disebabkan kerana ni effect dia okey dalam ni okey at this pH the attractive force between the positive and negatively charged group lead to precipitate of the protein molecules. Okay, in the lower pH okey keadaan berasid okey. So kita punya amine groups dan juga carboxyl group. So carboxyl group kita masih lagi ada H, so tak akan berlaku proses zwitterion. So zwitterion ataupun nama lain adalah isoelectric point atau PI. Zwitterion sama PI sama okey. Zwitterion inilah keadaan zwitterion di mana COH H dia tak ada. Negatif okey. So pH berada zwitterion okey. So this one kalau buat eksperimen dia nampak seperti cloudy sikit. Okay. So we put in high pH, alright. So kedua kita punya kumpulan amin dan juga kumpulan karboksil H dia akan hilang. Itulah prinsipal di sebaliknya. Okay. So daripada acid, daripada clear kepada cloudy, cloudy kepada precipitate. Okay. So cation, clear, cloudy, precipitate, anion. Okay. So there are several video yang boleh dapat daripada apa tu nota yang saya bagi baru ni. So boleh tengok video tu mengenai semua tentang test of proteins. Okay. Alright, so ada soalan sebelum kita tamatkan kita punya briefing hari ni? Is there any questions before saya lepaskan awak lagi sejam untuk memahami test of proteins? Sambil-sambil tu awak boleh jawab soalan ni. Okay. Ada soalan? Is there any questions?
So,
okay, yang lain ada? Ya.
Saya nak tanya.
Ya, saya tak dengar. Okay.
PK1 tu apa tu? PK